Good evening, everyone. My name is Candace, and I'm an event manager at Town Hall. On behalf of Town Hall and the University of Washington, it's my pleasure to welcome you to tonight's live stream presentation featuring cutting-edge research by UW grad students Kevin Bishop, Bree Gable, and Gabriella Reggiano. This is the second of 6 in our series, UW Engage Science, and we are so pleased to be able to offer this platform to these students and their important and engaging work. As we get underway, I would like to acknowledge that our institution stands on the unceded traditional territory of the Coast Salish people, particularly the Duwamish. We thank them for our continued use of the natural resources of their ancestral homeland. Thank you for tuning in tonight. Town Hall is proud to be a community-focused organization and a place where we can share and sustain ideas and creativity, even if it means we can't gather in person. I'd like to thank tonight's speakers for appearing tonight to help make that possible. If you share in Town Hall's vision for a robust community engaged in the arts, science, and culture where everyone has a voice, please consider donating tonight or becoming a member. Town Hall is adding new programs every day. You can check out more of what's upcoming by visiting our online calendar at townhallseattle.org. Tonight's program will include 3 separate presentations with a time for questions at the end. Each presentation is about 20 minutes long. Questions will be selected from those in the chat field at the bottom of the video player, so please submit those at any time. You can also text questions to 206-504-2857 as noted in the chat. If you can be sure to address the question to the presenter you intend it for, that would be greatly appreciated. We cannot guarantee that we will get to every question, but we will try to get to as many as possible. For those who would like to view the program with closed captions, you can click the CC button in the bottom right corner of the video player. The program will be available for re-watching immediately following the event. Town Hall's work is made possible through your support and the support of our sponsors. Our Arno Matulski Science series is supported by Microsoft, KUOW, the Northcliffe Foundation, Wyncote Foundation Northwest, and the taxpayers of Washington. Finally, Town Hall is a member-supported organization, so I want to thank all the members watching tonight as well. And now I would like to introduce our first speaker. Kevin Bishop builds machines that shoot lasers at brains to make them glow, a key part of microscopes that scientists and doctors use to look at individual brain cells. His goal is to use light to make tools to treat brain diseases like cancer and Alzheimer's. Kevin is working to tackle the challenge of distinguishing between healthy brain cells and infected ones by developing a microscope that highlights individual cancer cells during surgery. Kevin is studying bioengineering and is from Portland. Please join me in welcoming Kevin Bishop.
Thanks so much. 12 months. Imagine being told that's how long you have to live. How would you feel? This is the reality for around 10,000 people who are diagnosed with glioblastoma every year in the United States. This is one of the most common brain tumors, and the median survival time for folks with high-grade gliomas, which is the category that this tumor falls into, is around 12 to 16 months. And this is devastating, right? Someone could go to the doctor with peculiar symptoms or maybe even no symptoms at all. And they do an MRI and look at the person's brain, and they find this tumor, and the doctor says you have around a year to live. And if we want to think about why this cancer is so deadly, part of the reason is because, at least so far, it's basically impossible to remove all the tumor from the brain. So even if surgeons can get most of the tumor out, there are always these tiny pieces that get left over in the brain, and those pieces grow back and cause the person to pass away. In other words, there's no cure. Hopefully one day we'll have a cure for this cancer, but in the meantime, what we're asking in my lab is, can we help people live longer? Could we give people more time to spend with their loved ones before their cancer grows back? And it turns out we can help in kind of an unusual sounding way, and that's by shooting lasers at people's brains. And this sounds like a strange idea, like something out of Star Wars, or maybe even a horrible idea. But this is actually a way that we can help treat brain cancer. And I should be clear about something here. I'm not personally shooting lasers at anyone's brain, and trust me, that is better for everybody, because I'm an engineer and I don't have any business doing anything to anyone's brain. But I am building tools that surgeons can use to shoot lasers at brains, which can help them to find cancer and remove it more easily. To back up a second though, I've been talking a lot about surgery, and the reason is because usually surgery is the first step to treating these tumors. And really the goal of the surgery is to remove as much of the tumor from the brain as possible. Because removing more of the tumor can help people live longer before the tumor grows back. Think of it kind of like weeding your garden. Maybe you can't get all the weeds out of your garden. Maybe there's always one or two that gets left over. But the more of those weeds you can remove, the longer it's going to be before they all grow back. The same kind of idea is true for brain tumors. Surgeons are never able to remove all the tumor for this type of cancer, but the more they can remove, the better chance someone has of living a long time. Now, to be clear, there's a lot of different things that impact how long a person survives, probably even including things that we don't understand yet. However, the amount of tumor left over seems to be a big factor. Removing these tumors is challenging though, and to illustrate why, I'm going to take a minute to tell you a story, and that is the story of Kevin the Unreasonable Dinner Guest. So imagine it's the after times and you're having a dinner party, and everyone comes over to your house and gathers around the table. And unfortunately for you, one of those people is Kevin. And you start things off with an appetizer, and you pull out this beautiful block of blue cheese, right? That's this white cheese with all these moldy blue crevices running through it. And some people don't like blue cheese, and that's totally fine. But unfortunately for you, Kevin is one of those people who doesn't like blue cheese. And Kevin, being the unreasonable guy that he is, looks at the cheese and he says, "This is disgusting. This has mold in it. I can't eat this." And he says, "I want you to cut all the mold out of my cheese." And you look at Kevin and you look at the cheese and you think this is kind of a silly request, but you're a very gracious host and you don't want to make a scene. So you take your cheese and you take it back to the kitchen and you start trying to cut the mold out. And this is tricky, right? Because the mold is not in one spot that you can just cut away. It's sort of spread all throughout the cheese. But maybe if you're very slow and very careful, you can get most of the mold out. So after a few minutes, you take it back to Kevin and you say, "Kevin, here's your cheese, mold-free, let's eat." And Kevin looks at the cheese and Kevin says, "You know, actually I've changed my mind." And Kevin says, "I've brought my own cheese." And he whips out this other block of cheese. And he says, "This cheese is much like your blue cheese, but instead of blue mold, it has white mold in it." And Kevin says, "I still don't want to eat the mold though. So I want you to cut the mold out of my block of cheese. And you look at Kevin and you look at the cheese and you look at all your other dinner guests kind of staring awkwardly on as this situation unfolds. And you say, Kevin, I think you're just going to have to eat the cheese because there's basically no way that I can get the mold out of here. The mold is still spread all throughout the cheese, but now I can't even tell which part is mold and which part is cheese because they look almost the same. And at this, Kevin becomes very angry and he says, this was a very expensive block of cheese, which I brought to your dinner party and I want to eat it. And I don't want you to waste any of it. So if you accidentally cut out any of the normal cheese when you're removing the mold, Kevin says, I'm going to cut out a chunk of your brain. And hopefully no one has ever been in a situation remotely similar to this. But this is actually not that different from what doing these brain surgeries is like. These tumors are spread all throughout the brain. They look pretty similar to the rest of the brain. And if the surgeon accidentally removes part of the healthy brain while they're doing surgery, that can have serious problems for the patient. And what this means then is that even good surgeons can miss pieces of cancer. And I'm not saying that any surgeon is able to get all the cancer out. That's not something we can do at this point. But what I mean is that sometimes there are pieces of cancer that could have been removed if the surgeon could see where they were, but the surgeon couldn't see them, so those pieces got left. And what this would look like is if we imagine someone's brain and all these little gray dots are pieces of tumor. And ideally what would happen is the surgeon would cut out all these pieces. But the reality that a lot of patients face is the surgeon is able to get this main piece of tumor out, but these edge pieces get left over. And those are pieces that could have been removed, and those are pieces that grow back more quickly. This is an MRI taken of someone's brain that had basically this exact same thing happen to them. And this was taken right after the person had surgery to remove the main piece of cancer from their brain. And this MRI shows a little piece of missed cancer. And this is something that the surgeon maybe could have removed if they had been able to see it. And if we look at how time progresses after the surgery, we can see that piece starts to grow back. That white area expands around the brain, and that's the cancer coming back, and that would cause someone to pass away. If the surgeon had been able to get that piece out, maybe this person would have lived a little longer. Fortunately, though, folks have come up with something that's called the pink drink. And it's not pink, but patients do drink it, and they drink it before they go into surgery. And then what this does is when a patient has had the pink drink, when they go into the operating room and the surgeon opens up their brain, they shine a purple light on the brain, and that makes all the cancer in the brain glow pink, just like a glow stick. So content warning, this next slide has some pictures of brains on it. So if you don't want to see that, feel free to look away. That's totally fine, and I'll tell you when it's safe. But on the left here, we have a normal view of the brain. So this is what the surgeon would see sort of in the operating room under normal room lights. And in this left view, it's pretty hard to tell which part is tumor and which part is healthy brain. On the right, though, we can see the result of the pink drink. We see this purple light shining down, and we see that bright pink glow in the middle, and that's the tumor. And with this, then the surgeon can see very easily where the tumor is, and they know where to cut and where to remove. Okay, so it's safe now. You can look back, no more brain photos. And this kind of shows what's going on step by step. So we have some tumor in the brain that's hard to see, and the surgeon shines their purple light on it. The tumor glows pink, and then the surgeon can cut it out. And this sounds great, right? And this actually works pretty well for many aggressive tumors, and it's now done pretty routinely. And this can help surgeons remove more of the tumor. But there is still a problem. And that problem is that some pieces of tumor don't glow bright enough to see. It's kind of like when you've had your glow stick for a week and it's been hanging out under your bed, and you pull it out and maybe it's still technically glowing, but it's too dim for you to actually see the glow at all. And there's two main reasons that this kind of thing can happen to tumors. The first one is that oftentimes less aggressive tumors don't glow as brightly. And when I say less aggressive, I just mean that these tumors are not growing as quickly. And that doesn't mean they're good. It still means they'll eventually progress and cause problems for the patient. So we still do need a way to remove them. But what this would look like is there'd be some tumor in the brain that's hard to see, and we shine our purple light and we're ready for something to happen, and nothing happens. The glow is too dim for the surgeon to see it with their naked eye. The other thing that can happen sometimes is small leftover pieces that are in the brain sometimes don't glow as brightly. And what this might look like is we're able to remove the main piece of the tumor from the brain, this bright pink glowing piece. But there are these leftover pieces that get left behind, and those are pieces that can grow back. And those pieces are too small for the surgeon to see. And maybe you've noticed that I've been drawing the brain as the tumor rather, as this nice circle up until now. And that's not very blue cheese-like. And in reality, this is really where this blue cheese idea comes in, because oftentimes after that main tumor is removed, these side pieces extend away from the main tumor. And these are the parts that are hard to see because they're so small and they're hard to remove. So what can we do if we have things that are too small to see with the naked eye? Well, we need a microscope. And right, a microscope is just something that takes something that's small and hard to see and it makes it big and easy to see. So we can put a hair or a leaf or whatever else under here and look at it, and it shows us all these details that we couldn't see by just looking at the thing. But there is a problem with using this approach during brain surgery. And it's really more of a practical problem rather than some fundamental problem with using a microscope. But that problem is that you can't put someone's head under a regular microscope. So if I take someone's head, in this case, a picture of my face, and I try and shove it under this microscope, that doesn't work. And it's not just a matter of getting a microscope with more room underneath, because in order to make a very high resolution microscope, we have to put whatever thing we're looking at right underneath the microscope. And if I'm having surgery done to me and my head has been cut open, I don't really want you to be moving my head around to get it in the right position. So we need a different strategy. And this is a solution that my lab has been working on. And this potential solution is a handheld microscope. This is something that surgeons could hold, kind of like a pen, really big pen, but still kind of like a pen. And it maybe doesn't look like a microscope in the last slide, but it's the same idea. We have a miniature camera in the back, and then we have lenses at the front that magnify things. And then there's also a laser that gets shot out at the front. And this is where we get to shoot our lasers at brains. And this purple laser is what provides that purple light to make the pink drink glow. And surgeons are then able to hold this device right up to the brain, and it will show them those dim pieces of cancer that are too dim to see with the naked eye. And they can't put their face right up and look through this microscope like you would sort of a standard version. So the view gets put on this TV screen in the operating room. And what a surgeon might do is there'd be some area of the brain that looks healthy to the naked eye, and they want to check it out. So they take their microscope and they put it up against that area. And maybe the TV screen shows nothing. So that might indicate to the surgeon that there's no cancer there and that area really is healthy and they should leave it alone. But say they check out another area and that area lights up with all these bright spots. Those glowing spots would be the pink drink lighting up. And they know that this is an area that has a lot of cancer and that area should be removed. So this microscope can help surgeons see tumor cells that are invisible to the naked eye during surgery. And the reason that during surgery part is so important is because I've been showing you all these images so far of pieces of leftover tumor on MRIs. But the thing about MRIs is we can only take those after the surgery is over. And at that point, it's too late to do anything about it. But what we could hopefully do with this microscope is we could give surgeons a way to find those leftover pieces during surgery, and then they could remove them and help people to live longer. And my project is really to refine this design so that it's ready to be tested in patients. So this microscope, this device that I've shown you in the last couple slides, was developed by a few past grad students in my lab. And they came up with this great initial design, initial device, and they gave it to surgeons for the surgeons to test out. And the surgeons said, you know, this is great. And we need one main improvement before we can try this out in patients. And that's that we need to see a larger area at once. And kind of their logic there was that the surgeons think that, you know, maybe it's nice to be able to see individual cells in such high detail, but we don't actually need that much detail for what we're doing. And they'd rather see a bigger area of the brain. And to sort of motivate where the surgeons are coming from, I took this video of my coffee mug. And this video is very zoomed in. So it's very detailed, very high resolution. And maybe I can see all the details on each individual letter. I can see sort of the border of each letter, and I can see the dots on the eyes. But because the viewing area is so small, it's pretty hard to read this sentence. I can't see the whole thing at once, and sort of because I'm holding my phone in my hand, just like the surgeons would be with this microscope, any small movement of my hand becomes really obvious and makes it challenging to read things. But if I then take another video, and this one's zoomed out, so there's less detail, there's lower resolution, but I have a larger viewing area, even though I can't make out as much detail, I can actually read the sentence a lot better because I can see the whole thing and these movements aren't nearly as noticeable. And this is what surgeons were getting at, because really the same idea applies to cancer tissues. So I'm showing you here 2 images of brain, and these images are very zoomed in. This is an image that you would get like from our microscope. And on the top is cancerous brain tissue, and on the bottom is healthy tissue. And you can sort of make out these very fine details. Each of those bright pieces is a cell. But these 2 views look pretty similar. So in terms of telling cancer apart from healthy tissue, this view makes it pretty challenging. But if we then zoom out, so this green box on the right shows the small zoomed in area that we were looking at on the left. This larger viewing area can help surgeons see what's going on. And we can see that the structure of the cancer in the healthy tissue actually looks pretty different. The cancer has a lot more of these bright spots. They sort of have this swirly, chaotic pattern, and the healthy tissue doesn't. So being able to see more of the tissue, even if we have lower resolution, can actually be more useful. And a larger viewing area is a little easier said than done. It's doable, but it's tricky. And that's kind of why I'm spending so much of my research work trying to make this happen. And the reason it's tricky is because to get a larger viewing area, we have to change the lenses that are inside this microscope. And because this is a miniature microscope, it's something you can hold in your hand, we can't just go to the store and buy lenses off the shelf that work and pop them into place and call it a day. Everything has to be custom manufactured and custom designed. So I'm working with a company that really specializes in making these miniature lenses to develop a new system of lenses that will allow us to have a bigger viewing area. And then hopefully with that new device, we'll be able to get this into the hands of surgeons and they can test it out in patients. Even if our device can't ultimately cure this cancer, our hope is that it could increase survival time and give patients longer before the cancer grows back. Say someone lives an extra 6 months. That doesn't sound like much, but if it's your last 6 months, that could be invaluable. Maybe someone gets to witness their child's wedding or hold their grandchild for the first time, or just spend that much more time with the people that they love. That could be truly life-changing.
All right, thank you so much, Kevin. I want to remind folks in the audience listening that you can submit questions at any time. So if you have questions for Kevin, submit them now before you forget. All right, so our next speaker is Bree Gable. An experienced aquaculturalist, Bree managed a community science team for nearly a decade, focusing on rearing jellyfish for aquarium display and scientific research. For the last 2 years, her jellyfish have been used to help scientists understand what jellyfish are eating and how that might influence the entire Puget Sound food web. Bree is studying museology and is from Seattle. Please join me in welcoming Bree Gable.
Thank you so much for that introduction. Today I will be talking smack, teaching you what jellyfish have to say. There we go. So if you're like me, you've probably spent a lot of the last year scrolling on news feeds, probably more doom scrolling than we all should be doing. And this is what my feed tends to look like. A lot of articles about jellyfish, specifically jellyfish taking over the world. And at first glance, that's pretty alarming. No one wants their world taken over by jellyfish, but in reality, they are not taking over the world. But smacks of jellyfish, groups of jellyfish, are becoming more frequent. So they aren't taking over. What are they doing then? When I see articles like this, I see misunderstandings or miscommunications between jellyfish and humans. And that's because I've spent a long time learning to speak jellyfish. I'm a jellyfish aquaculturist, which basically means I grow jellyfish. These jellyfish can be used by scientists to help answer their questions, or they can be used to educate people about jellyfish, about Puget Sound, anything that has anything to do with jellyfish. And because of that, I have a lot of interesting insights into what the presence of jellyfish might mean when they're taking over. And a lot of that comes from understanding this right here. This is the life cycle of a moon jellyfish. And it's more than likely that you probably never thought about where jellyfish come from. I think about it a lot. I spend so much time thinking about this. And it's because I understand this that I can kind of tell you where some of these miscommunications might be happening and how these increased jellyfish sightings might actually influence the larger ecosystem at hand. So let's take a minute and actually go through this life cycle so we can all get a better understanding of it. The part of the life cycle you are most probably familiar with is this stage right here called Medusa. These large free-swimming jellies kind of look like the head of the mythological creature, which is where they get their name from. But at this size, which is about the size of a dinner plate, you can actually tell if they're male or female. Females will typically have this pinkish color in the center of these horseshoe-shaped things, and males will have a whitish color. And the whole job of these Medusa is to eat, to get big enough to breed. And when they're at that size, that big dinner plate size floating around, the males release strings of sperm, which I hope you can see here on this picture, in hopes that the females will come by, and as they pulse, they swoop up those sperm strings and fertilize their eggs. The females fertilize their eggs and actually hang on to them for a few days as those early cell divisions occur. And after a few days, these little baby embryo jellies, not really jellies, become planula. Planula are these microscopic little balls covered in hairs, and they have one job: get to something hard. So after those couple of days, the female pulses and swoops her arms, and those planula are released into the water. Most of them get eaten by other animals, but every once in a while, one of them makes it to that perfect spot and transforms into a polyp. Now, these polyps are also really, really small, think size of a grain of rice. But if we look closely, you might see some things that are familiar to those Medusa stages. You can see tentacles, right? And in the center here, there's that mouth, kind of like in the center of those other jellyfish that we typically see. But they're stuck to rocks. If you are familiar with jellies, you might know that they are cousins of corals and anemones. They belong to a big group of animals called cnidarians. And that's why these guys kind of look like they're cousins, because they're all kind of related in that fun way. Now, these polyps have one job, two jobs really. They can either divide and make more of themselves. Like I mentioned, they're related to anemones. So here you can actually see an anemone stretching out and splitting off to clone itself. The polyps can do essentially the same thing. They can stretch out and pinch off buds to make more of them. So a single planula can make one polyp, which can then make thousands of other polyps. And they'll basically stay that way unless something comes along and eats them or something disturbs them. The other thing they can do is strobilate. And this is the process in which we get our jellyfish from. So rather than cloning themselves, the polyp pulls in its tentacles and creates a stack of pancakes. And as you move from the bottom upwards, the little jellyfish become more developed. At the very end, they eventually start pulsing, and then they pop off and swim away into the water. These little things are called ephyra. They're typically reddish in color, again, really small, think size of a grain of rice or smaller. And from this point on, their whole job is to grow up. And after about 4-ish weeks, they're typically about quarter size. They slowly lose that red color and become transparent and become those adult Medusae that we started with. And at this point, their whole job is just to repeat that cycle, get big and make more polyps, or more planula that become polyps. So now that we understand this life cycle a little bit better, we can kind of start to interpret what it means when we see jellyfish. If we're seeing lots of jellyfish here, it probably means something is going on in that life stage that we don't normally see. Maybe there's just a lot of polyps because we're building things that polyps like to live on, or we're actually triggering the polyps to strobilate. Now, as a jellyfish aquaculturist, I take advantage of this. I intentionally subject my polyps to temperature swings and salinity changes, how salty the water is. So it basically tricks them into thinking it's time to strobilate. This is also happening due to climate change. So it's not that jellies are taking over the world, they're just reacting to their environment. And we can tell a lot what's going on in their environment by just them being there. So here's a picture of a smack of jellyfish in the wild. Now, I have been doing this for so long that I can actually interpret what the jellyfish are saying by just looking at the picture. I can tell you that it's probably a little bit later in the year. The jellyfish are pretty big, so they're probably about to spawn really soon. If not, they probably have already spawned. And the water's pretty green and pretty murky, which makes me think it's probably late summer because that's when we get lots of sun and lots of algae growing in the water. And the smack itself is not super dense. And I also don't see any other kinds of jellyfish in the water, like egg yolk jellyfish or lion's mane jellyfish. These jellyfish actually eat moon jellies. And the reason I think that it's a little bit warmer is because I know these jellies prefer a little bit colder water, having tried to grow them myself. So just looking at these things and being observant, I can kind of tell you what's going on in Puget Sound by just what the jellyfish are doing there. But what does that really mean for everything else, right? If there's more jellies and their whole job is to grow big, they got to be eating something. Well, what are the jellyfish eating? Puget Sound is full of plankton, microscopic animals and algae that make up the base of the food web. If you eat seafood, at some point that seafood ate plankton. And I want you to think of this plankton as trail mix. There's some good stuff, there's some bad stuff. I like M&Ms. I do not like raisins. So the plankton are kind of the same way. We've got some M&Ms and we've got some raisins. The M&Ms are typically small microscopic animals. Like this guy right here is called a zoea. He actually grows up and turns into a crab. These guys are called copepods. If you've ever watched SpongeBob SquarePants, this guy right here is what plankton, SpongeBob's nemesis, is named and designed after, or he's designed after him. He is a plankton, right? Pretty cool, huh? And then the not so good things are typically algae. Now, it's not to say that nothing eats the algae, but if you have a choice between raisins and M&Ms, I'm going to probably choose the M&Ms. So if the water conditions are making more jellyfish, and these jellyfish whole job is to eat and make more jellies, what does that mean for everything else that also relies on this bowl of trail mix? Like the fish larvae that might grow up into becoming salmon, which then go on to support our resident orcas, or even us. I personally love salmon. Or does this increase in jellyfish actually benefit salmon? Because as we are learning, salmon actually eat jellyfish quite often. Oops, sorry, there we go. So that's the big question that we're trying to answer. How do jellyfish influence Puget Sound's food web? Now, this is a really, really big question. And unfortunately, scientists are not fluent enough to really answer it. You don't learn English by watching Shakespeare plays. You start asking really simple questions first, right? Hello, my name is Bree. My favorite food is M&Ms. That's the kind of thing we're doing right now, so we can become more fluent and understand what's happening in the larger picture. What do moon jellyfish eat? How much do they eat? Does the size of the jellyfish matter when it comes to all of this? Now, even though I said I could tell you a lot about what is going on in Puget Sound by looking at that, that's all anecdotal, right? That's just my personal observations. And even after almost 10 years of doing this, if I was taking a series of classes at UW called How to Speak Jelly, I would have just finished Jelly 101, and I'd probably be starting Jelly 102 relatively soon. Again, it's a big, complicated language that we're trying to learn here. So to answer that, I'm not the only one that wants to know. And it's always easier to study these kind of things with friends, which is why with a project like this, you need some study buddies. Of course, there's me, the person who grows the jellyfish, but I'm not an expert in plankton, so we have plankton experts. I'm not an ecologist, so we have ecologists like Dr. Corey Green, who is also our resident kayak expert, which we'll get to in a moment. And of course, all of this comes back as numbers, right? We need someone to crunch the math. So we also have people who are really good at that as well. And this is by no means the entirety of this group of people studying this. These are the people I worked most closely with. So I know them well enough that I can find their pictures on the internet to include them. And of course, all of this stuff needs some money behind it. So I want to give a special thanks to Sea Grant for funding this cool project. And I never liked growing jellyfish by myself. I think it's a fun thing to do as a group. So I want to also give a special shout out to the jelly team, the community science / aquaculturists who helped me actually grow the jellyfish that was used in these experiments last year. So once you have your study buddies, you need some tutors, right? You're learning a language. You might as well speak to the people who natively speak it. In this case, that's jellyfish. And you can either learn it in a classroom, like a lab, or you can learn it in the country itself, the field. Well, jellyfish, I'm basically a jellyfish babysitter at this point, and that meant I had to stay home and take care of the baby jellyfish. So I didn't get to do any of the cool study abroad stuff on this cool research ship. I got stuck at home taking care of the jellies, which I do love. I won't trash them too hard, but I'm going to be focusing more on what we did in the lab experiments to actually learn what the jellyfish are doing. So once you have your study buddies and your tutors, you just study, right? You do some experiments, try and figure out what's going on. And this looks crazy, and that's because it was crazy. It was the middle of summer. We had a whole bunch of water everywhere, jellies in yogurt cups. So I'm going to walk through kind of what we did because I think it's really cool. And it's an interesting way to learn how jellyfish or learn what jellyfish are trying to say. So we started off with 10 identical tanks. The reason we had 10 is we wanted to have duplicates, mostly because the more duplications you have, the more likely it is that you can, or less likely it is that you'll screw up your own science. There were times when we might have dropped a cup of plankton or sucked up a jellyfish or other mistakes that might have happened. So by having these duplicates, we were making sure not only our science was more secure, but getting that science was a little bit easier. So in 3 tanks, we had 2 jellies. In 3 more tanks, we had 4 jellies. And in the last 3, we kept empty to compare it to after a certain amount of time. And so the tanks themselves simulate that open water, right? Because the jellyfish, they're constantly living in that open water environment. So the water spins around and the jellies, all they got to do is eat. But if we're trying to figure out what they're eating, we have to go get some trail mix. Now, it would be really easy to just go to Costco and buy a bulk bag of trail mix. That's kind of what we did here. In this case, we went to Puget Sound, right? We went right off the dock and got some trail mix ourselves. The way we did that is we took a plankton net, which is basically a large filter sock with holes typically about the size of the width of your hair. And what that did was it filtered out animals that were bigger than those holes and let everything smaller go through. And by dragging that through the water, we condensed all that trail mix into a tiny amount. So one lucky person got to go out in a kayak and drop that plankton net in the water, and we pulled it back in with a crab pot puller because that thing got heavy really fast. When you're taking thousands of gallons of water, that thing gets heavy pretty quickly. And there's lots of stuff living in it that also made it harder to drag. Once that plankton net was pulled in, we would put it into a bucket. This is what Puget Sound trail mix looks like. It's brown and it's sludgy and it smells awful. Think a very hot summer low tide. Very, very stinky. And this right here is all the information we want, right? There's those cool zoea and those plankton copepods, and there are lots of raisins. Since we had 10 tanks, we then divided them into 10 little containers to put into those tanks. And you might be thinking, hey, I can see things. Those are raisins. The diatoms, those algae, they're kind of large and they're also kind of sticky. They're almost like Velcro. What you can't see are the M&Ms. All those little plankton, those copepods and zoea are swimming around. And again, they're so small, you just can't see them from right here. But the jellies know they're there. So we would add our jellies, we would add our plankton, and let the jellies do their thing. With every pulse, the jellyfish is swooping its tentacles through the water, trying to catch those plankton. And as it does that, it moves them into its mouth in the center. That way, that's how it eats. It's pretty cool. So after about 2 hours, we said, okay, jellies, your job is done. We took them out, put them in these incredibly scientific yogurt cups, and put them back in the main aquarium. Then we took essentially smaller plankton nets. These cones here, or these cups here, have plankton net in the bottom, and drained all the water out. Because what we did, instead of trying to figure out what the jellies had eaten, we looked at what they weren't eating, so we could work backwards, essentially. So after that, we have lots of cups and lots of samples, and the only way to figure out what's in them is to count the plankton. Now, this takes a really long time, and this is the step that we are currently at. And I say we kind of loosely because I'm not doing the plankton counting, and honestly, I'm okay with that. I like growing the jellyfish. I don't like being crouched all over a microscope all day looking at plankton. So this is kind of where my journey ends, but not really, because I'm an educator. Instead of just saying, okay, Bree, we're done with your jellies, we said, okay, Bree, you like talking to people? Do cool things like this talk and tell people about all the cool research we're doing and teach people how to speak jellies themselves or speak jelly themselves. So how can you do this? Well, like any good teacher, I should be assigning some homework, right? To do that, I should probably make you some homework. So what I have been working on is I have been working on tools to help people learn to speak jelly. And one of those tools is a video game. So hopefully very soon, you can actually play as a jellyfish and explore Puget Sound and collect that trail mix yourself and use that trail mix to unlock and learn about Puget Sound animals on the food web. That way, if you're not near Puget Sound or there's a pandemic going on, you can still safely learn this stuff without having to do anything crazy. Just open it up and have some fun. So now that we've actually had a chance to learn a little bit about how to speak jellyfish, I want to practice. Now, if you remember right, this picture here is the one I showed from before. So when you come across a smack of jellies in the wild or in an aquarium or wherever, I don't want you to think, oh my gosh, these jellyfish are taking over the world. I want you to look critically at the things around you. What's the weather like? What time of year is it? Are there any other animals in the water that might be eating the jellies or living amongst the jellies? Ask yourself these questions. Practice your observational skills. Because even though I say jellyfish can speak to us, in reality, all it is science. And science is based off observations. So the more you practice that, the more fluent you can become in jelly, but also just nature itself, which is really cool. Now, these jellies, if you listen really closely, I have actually been working with them a little bit longer, and I asked them to give you this special message, which is thank you so much for listening to my talk.
All right, thanks so much, Bree. I love knowing that a group of jellyfish is called a smack. That's pretty fun. All right, so our final speaker is Gabriella Reggiano. Gabriella works in computational structural biology lab, a computational structural biology lab, a fancy way of saying she spends her time at a computer writing a bunch of code and looking at a lot of protein models. Unlike most scientists, her goal isn't to answer a fundamental question about the universe, but to develop a method that will make it easier for scientists to understand how proteins move so they can answer whatever questions spark their curiosity, as long as they're about proteins. Gabriella studies biochemistry and is from Connecticut. Please join me in welcoming Gabriella Reggiano.
Okay, thanks, Candace. So today I wanted to talk to you about proteins, specifically proteins that don't behave the way scientists want them to. So when you hear protein, you might first think of nutritional protein that you need in your diet, or you might think of whey protein that you would put in your shake after or before your workout. I don't work out, so I don't really know when you're supposed to eat it. But what I'm actually talking about are these guys. This is a collage of proteins, small molecular structures in your body. And don't worry, I also think they look insane. Proteins form all kinds of crazy structures. And this is only a small fraction of the proteins that exist. Scientists estimate that you have at least 20,000 different proteins in your body. And the proteins in your body are responsible for all kinds of important jobs. And when those proteins stop working, our bodies have a lot of trouble doing everyday tasks that we take for granted. So this is a protein I studied in college that helps your heart pump blood. A patient had a version of this protein that was only 0.5% different. And if you notice on this picture, I've colored one tiny region of the protein a different color. And that's the only part of this protein that was different for this patient. But because of that small difference, they had a heart disease that could eventually cause their heart to stop pumping. So I knew how the protein was supposed to work without that little difference. And if I could understand why the patient's protein stopped working, maybe I could get it to work again. And that means that understanding how a protein does its job can help us fight disease. And proteins are responsible for all kinds of important jobs in our body. They send and receive messages, turn our food into energy, and contract our muscles so we can work out before or after drinking that really gross protein shake. They're essential to our everyday lives. And you can think of proteins kind of like the kitchen utensils in your kitchen. If we think of a chef in his kitchen, in his element, whatever he's deciding to make for dinner or dessert, he's got a bunch of kitchen tools at his disposal. If he wants to make a meringue, no problem. He's got a whisk that will add air to his eggs. Need to flip a burger? Fine, he's got a spatula. Want to serve some soup? No problem, here's his ladle. Each of the tools in our kitchen has a specific job. He wouldn't be able to use the spatula to serve soup to his customers. And our body is just like the chef. Only instead of kitchenware, our body has a fancy array of proteins, and each protein is responsible for a different job. And remember, if a protein can't do its job, there are serious consequences. Think about one of your favorite recipes to make at home. Maybe that's a pie or a curry. For me, it's probably spaghetti and meatballs, partially because I'm a lazy cook and partially because I'm Italian and I like to eat pasta. Imagine you forgot to wash one of the tools that you need to make your dish. Suddenly, it's a lot harder to whip up that meal. If I don't have my tongs, how would I get my spaghetti out of the pot? Am I gonna fish out each of the noodles with a fork? That's much harder. The same idea goes for proteins. If a protein stops doing its job, some of these essential functions may not be able to happen. And if we want to bring that function back, we need to know how a protein did its job before it broke down. But there are a lot of proteins. Remember, we have well over 20,000 just in our bodies, and proteins are also in plants and animals, bacteria, and viruses. Luckily for us, all we need to know to understand how a protein does its job is to figure out what it looks like. Think back to our kitchen tools. Our spatula is really good at flipping burgers and not so good at serving soup. But what makes it good at flipping burgers? It's flat so it can get underneath. It's wide so that it doesn't break the burger when we flip it up, and it's got a long handle so we don't burn our hand. The shape of the spatula is essential to its burger flipping prowess. So then all we need to understand what a protein does is to know what it looks like. If we can figure out its shape, we can understand what job it does in our body. So this is an image of a protein, one where we look down on it from a bird's eye view, and another where we look at the side. And it looks pretty crazy. It's got all of these bumps and pieces to it, but let's focus on its general shape. It's got a big hole through the center. So you can think of this protein like a pipe. It just lets other things in our body move through it. But unlike a pipe, proteins are really, really small. If we were to look at this protein and measure its diameter, it would be about 85 nanometers long. So 1 nanometer times 1 billion is a meter, and 1 meter is equal to 3 yards. But this billion is such a large number that it kind of loses its meaning. So for context, a sheet of paper is 75,000 nanometers thick. And that means our protein pipe is 1,000 times smaller than the thickness of a sheet of paper. That's so small that if we put proteins under a microscope like this, the type that you might have used in your high school biology class, you still wouldn't be able to see anything. So if we can't see it with this microscope, how the hell am I even showing you this image? And even more than that, how does this image have details? How do I know that this piece down here is placed next to this piece right here? And how do I even know that these are two separate pieces? And remember, the details do matter. A 0.5% difference in a particular protein could shorten a patient's lifespan. And this level of detail requires a lot of power. So this is the microscope that we use to get those really detailed images. It's called an electron microscope because we use electrons to see microscopic things. And electrons are the negatively charged pieces of an atom. And it's because they're so tiny that they give us the ability to see very, very small things. And even though these electrons are very tiny, this microscope is not. For reference, this is around 12 feet tall. And I know you can't see how tall I am over Zoom, but the answer is very short. And next to this microscope, I managed to look even shorter. Before we put our protein in this very tall microscope, we need to do a little bit of prep first. We take a single drop of our protein sample, and that single drop will have millions upon millions of copies of our protein, and then we freeze it. After that, we can put it in that huge machine and shoot it with electrons. So all of the different copies of our protein are frozen here, hanging out, and then it gets shot with the electron beam. That beam is going to come down and turn all of our 3D proteins into 2D shadows. So if the protein is pointing up, we'll get a 2D shadow of what the top looks like. If it's laying on its side, we'll get a 2D shadow of what the side looks like. And it's really important that we get shadows of every side because proteins don't exist in 2 dimensions. Just like us, they form 3D shapes. So let's take a toilet paper roll and shine a light on it. Here we get a donut-like shadow. And if this was the only shadow we could see, this toilet paper could be a lot of things. It could be a donut or a bagel, even a flat CD. But if we get multiple views of the toilet paper, we can better understand its 3D shape. So after the computer collects all of our 2D shadows for us, we can try and use those shadows to get our 3D image. So what we can do is combine all the shadows that match each other, and this gets us an even clearer 2D shadow. And you can see the images here are a little more sharp than the images to the left. And then we can use those different shadows to get us back to the 3D image, to that picture of the pipe protein that I showed you before. And that model will have all of those details because we took hundreds upon thousands of images and combined all of them. This combination step is really important. For most proteins, it's the combination step that gets us the details that we need. But for some proteins, this combination step can actually erase some of the details. So I'm going to play a video of a protein here. It'll be a little choppy because of my internet, but that's okay. And this protein, pyruvate dehydrogenase, is just going about its everyday business. This is what it's doing in your body. And this movie is an artist's rendition of how this particular protein does its job. And just look at all of this motion. How could we ever hope to get a picture of something that's moving around this much? Think of that kid who won't sit still on picture day. It's a lot harder to get a clear picture of him. So if a protein is moving around a lot, then this combination step will actually erase some of the details that we need. And a lot of proteins are flexible. Many of them bend or change their shape in order to do their job. So if we can't get a clear image and a clear idea of how these proteins are moving, we won't know how to fix them if they break down. So to simplify the problem, let's talk about how this would work with 2D pictures. So here I've got a bunch of different snapshots of a Pegasus running, and you can see the legs are moving all over the place in these different images. One, we have all feet off the ground, and another, it's front feet on the ground. So just like we have to combine all the images we get under the microscope, we need to combine all of these snapshots into a single image. This is an essential, unskippable step with the microscope, so I have to put it in the analogy. So let's say our computer breaks, and whenever I try to show these images to my friends, the computer shows them this instead. An image that's a combination of all those snapshots we had before. In the snapshots, the legs were moving all over the place, so we don't get a clear image of them when we combine them. The legs are just a gray blur instead. The body and the head, though, didn't move as much, so those are a bit clearer. If we want to understand how the Pegasus is running, that's really difficult from this blurry image. How can I do that when my computer is only showing this image? Well, it turns out my computer will play movies okay, so what if we could turn this blurry image into a stop motion movie instead? So we don't know how a Pegasus runs down the street, but we do know how horses run. So we could use our knowledge of horses to make guesses about how the Pegasus might be running. So I know, for example, that a horse lifts all of its feet off the ground, so we could draw that in. And one time I saw a horse with 3 of its feet on the ground, so we can draw that in as well. And we can apply this same concept to proteins, using what we know about one protein to make guesses about another. A Pegasus has really similar features to a horse, though. If we think back to this collage of proteins, all of the proteins look completely different. How could we possibly use the information about one protein to inform us about another? Luckily for us, all proteins are made of the same exact building blocks called amino acids. And just like toy blocks, amino acids have different features. So here we have a triangle block, some rectangles, and even an arch that could go in our building. We could, for example, make a building like this, although it probably wouldn't last long. We put the triangle on the bottom when it has a pointy top that doesn't provide good support. We tilted the arch when it's heavy and needs more support. But we could also build a tower like this, which would stay put without us needing to hold it up. The triangle block goes on the top where it makes a nice roof. The arch goes on the bottom where it can provide more support as part of the foundation. So amino acids also have different features, just like these blocks. Some amino acids are sticky and they want to be around other sticky amino acids. Some are wet and they like to be around water. Others are attracted to each other and they want to be close. And some don't like each other and they want to be far apart. And so just like our toy blocks, those different features will determine the most stable arrangement for any protein. And there are 20 amino acids that make up every protein and have some of these features listed here. And that means that we can use these amino acid features in our understanding of how they like to fit together to make guesses about proteins. So just like we did with the horse and the Pegasus, we can apply our knowledge of amino acids to any protein we might be interested in. So that means we could take a protein and make an educated guess at how it might be moving. So this protein might bend to open it up like a mouth. So we've made guesses at how something might look, but then what do we do with our guesses? Let's go back to that Pegasus example. So we made guesses about our Pegasus, and now we can go and check if those were correct. We can go into the hard drive of my computer and we can look frame by frame and check if we had the right idea. So we can see we had one guess correct with all of the feet off the ground, but we had one guess wrong. We put 3 feet on the ground when there only should have been 2. And we can keep going through this process, making guesses at how the Pegasus might be running based off of our knowledge of horses, and then we can check if we were right in the individual snapshots. And if we can find snapshots that match our guesses, we might be able to turn that blurry image into something more informative, a stop motion movie instead. We could get a better, not perfect, but a better understanding of how that Pegasus is running down the street. So for a protein, we would take our guess at how the protein is moving, and we can hit it with a pretend electron beam on the computer, and we can create fake shadows. And we can do that for all of the different views of our protein. And then we can take our fake shadows and we can compare them against what we saw in the microscope. And maybe our guesses match, and that's great. But then we can keep doing this. We can make guesses and check them and see if some were right and if some were wrong. And then hopefully, just like the Pegasus, we can get to this point where we would have a stop motion movie of how the protein is moving. So these stop motion movies would give us more information than a blurry 3D image. But what would that information actually allow us to do? I wanted to spend time with a specific example so we could really visualize the importance. So this protein here helps malaria infect our bodies. And scientists found 2 different snapshots of this protein. And you can think of this protein kind of like an arm in tug of war. In this first image, it reaches out its hand and grabs onto the cargo. And then in the second image, it pulls the cargo down. But scientists don't understand how it moves from the first image, hand latched onto the cargo, to the second one, where it pulls the cargo down. And that's a lot of missing information. How can we stop this protein and thus stop malaria from infecting us if we don't understand how this protein is doing its job? So imagine someone keeps slapping you on the face, back and forth, and it happens so quick you have no idea how they keep slapping you. All you see is them standing there with their hands at their side, and then boom, hand on your cheek. And you've only got these 2 data points, hand at their side, hand against your cheek. How would you be able to stop them from hitting you when you can't see how they're doing it? You could try and make guesses about how exactly they're hitting you, but that's much harder and requires you getting hit a bunch of times before you figure it out. If their movement wasn't happening so fast, if it wasn't a complete blur and surprise to your eyes, you would have a better idea of how to stop them. And we want to get that same extra information from proteins that move. If we could get more data points about how this protein brings in the bad cargo, we would have more information at our disposal to figure out how to stop it. For any disease that can be traced to malfunctioning proteins, an understanding of how it moves gives scientists more information to work with, and more information will give us a better chance at fighting that disease. So with the method I'm developing, that would be a possibility for any scientist who's interested in a protein. And that would mean that movies like this one would be possible for any scientist. And hopefully, with my method, they wouldn't just be an artist's idea of how a protein is moving. They would be descriptions of what is actually happening in our body every day. And those descriptions could be vital for scientists who want to fight heart disease, prevent malaria infections, or understand any disease that might be caused by a malfunctioning protein. Thanks.
All right, thanks so much. I'd like to invite everybody on screen so we can get to some questions. You are still able, audience, to submit questions, so feel free to do that at any time. We are going to start with Kevin. So, Kevin, first question. What is the pink drink interacting with that highlights the tumor? What makes it highlight the tumor and not anything else in the brain?
Yeah, it's a good question. So there's a process, like a chain reaction kind of chemicals that all cells do, but cancer cells, it turns out, carry out this chain reaction, and they kind of get stuck at one point along there. And what the pink drink does is we basically give the patient a whole bunch of the starting point of that chain reaction, and all the normal cells just process that chemical and it gets all the way to the end. But for the cancer cells, that chemical gets stuck halfway through. And that middle step, that sort of intermediate chemical, is what glows. So they start to accumulate all this glowing chemical in the middle, and that makes the cancer cells light up.
Let's see. When you go to the zoomed out view, do you need to change the laser illumination area to compensate?
Yeah, another good question. So yeah, we do kind of have to redesign the laser, sort of how we light things up to go along with that. Basically, we're sending the laser in and we're making it into a line, so we can sort of highlight a whole line of tissue at once. So we need to make sure that line is sort of compatible with the viewing area we're looking at.
And you talked, kind of specifically just about brain cancer, but would this be a thing that would be used in other cancers, or does brain cancer kind of function and grow differently than other cancers?
Yeah, I mean, I think the general idea of having a microscope that a surgeon can use and get high resolution while they're doing surgery definitely makes sense for other cancers. I think the advantage with the pink drink is that's something that's already used for brain cancers right now. So we sort of have this tool that's already used in the clinic, and we want to take advantage of it by building a microscope that can look at it better. But there's a lot of other situations where it would be helpful to be able to get more detail during surgery. Breast cancer is another example where surgeons kind of have to make sure they get enough of the tumor out in order to not have to go back and remove more tumor later. So if there's a microscope they could use that could help them get a better idea of how much tumor they need to remove or how much tissue they need to remove, then that would also be very helpful.
And in my intro, I mentioned that you're using these also for Alzheimer's. Can you talk about how that would work? And like very briefly, it's probably very complicated, but how
sure.
different is that?
Yeah, so it's also a microscope, but it's a different microscope. So the microscope that I described is just for brain cancer. And then we're putting together another microscope that's more like a conventional microscope that kind of sits on a table. And that microscope basically is able to take 3D pictures of brain samples. And that would be more the sort of thing where someone who has Alzheimer's donates their brain, and after they pass away, we're working on something that can look at their brain in a lot of detail and maybe give us clues as to what's going on with the disease.
And then last question, how do you test a tool like this before it's used in a real surgery? What do you test it on to know that it's ready to go?
Yeah, the testing is kind of an interesting, I don't know, maybe ethical question, because we do testing on sort of samples of tissue in the lab, and we can look at them and see if we get a piece of cancer tissue from someone and see if it lights up. But then sort of the question that comes after that is eventually you have to test it on a person, and how can you do that? And usually what we end up doing is, with a device like this, we would sort of use this on a person, but we wouldn't tell the doctor what the result is, because we don't know for sure that our device is sort of giving us accurate information. And then the researchers would keep that information and say, like, okay, based on this device, we think that they missed a piece of cancer here. And then if later it turns out that we were right, then we know that the device is working well. But it's also possible that we could be wrong. So that's why you don't want to sort of communicate that information to the clinicians until you are more confident that things are working well.
Well, thank you so much for your presentation. That's fascinating, and we all know people that have been affected by Alzheimer's and cancer. So thank you for that important work. So let's see, now moving on to Bree. Yours was just so fun. Who doesn't love jellyfish? I grew up in a place where we didn't have any jellyfish, and I just go out now and I see them. So are you seeing that there are actually too many jellyfish in Puget Sound right now? And why, if so, why is that a problem?
So that's not really a question we can answer yet, because to say what too many is a question that would require years and years and years of research to really answer that. But I can tell you it changes from year to year. There are some years when all I see is moon jellies, and there are other years when I will see no moon jellies. In fact, last year, so this project happened over 2 summers, so 2020 and 2019, and last year they had a really hard time finding moon jellies, and so they're out on that big research ship looking for jellies and couldn't really find any. So it's hard to say because it does fluctuate so much. That, and we don't really understand yet who takes advantages of the jellyfish being there, like what really eats them. So that question is certainly something that hopefully one day we answer, but I can't give you any sort of concrete answer now. Ask me 50, 100 years when, we have a decade or a century of science, maybe.
Okay. Let's see. Why are strobila red while polyps and medusa, I'm probably murdering the pronunciation of these, are white and transparent or transparent?
So I don't have a specific answer for that. It could be a couple different things. It's not uncommon to see animals that are trying to stay hidden, especially in the water, to be red, because the way the light interacts with the water, red is actually absorbed very quickly. So if you see underwater pictures that are really washed out of all that red color, then that might be why, or that's because of the red light being absorbed into the water. So I would suspect that if it is red, it might have something to do with that. It could also have something to do with whatever kind of, because we talked so much about proteins today. There could be a special protein in there that is more red. I'm not actually sure if anyone's researched that specifically, but it's a very interesting question. Way to give me more research ideas.
You're welcome. Let's see. Can you talk a little bit, this is my question. I'm just curious, what kind of a brain does a jellyfish have? Like, what is it, and maybe where is it?
Okay, yeah. So jellyfish do not have brains.
They don't have brains.
They don't have any brains. They basically have a thin net of nerves scattered throughout that whole gooey part, the bell. And all that does is react to signaling, which is on, contract, or don't contract. So that's why they kind of do that initial pulse, which is really incredible because they've been on the planet for their at least, oh my gosh, 450 million years? Yeah, about 450 million years. And then with no brain, and here they still are, taking over the world.
Yeah, right.
Yeah.
Yeah. Okay, so last question. What should we do when we see jellyfish in the water? Should we interact with them at all? Do they all sting, or should we leave them alone?
So all jellyfish do sting. That is a characteristic of that large group of cnidarians. So all anemones, corals, jellyfish, they all sting. It's just a matter of, can the jellyfish, the stingers, essentially get through your skin to cause problems. It also depends on the individual. Yeah, I actually know people who have worked with jellyfish for a long time who've developed allergies to jellies, so they don't want to go anywhere near them. If you do see a jelly, I recommend just watching it, enjoying it. Don't touch it, because even after they wash up on the beach or something, they can still sting you. At least within Puget Sound, we don't have any jellies that will kill you, but they're not pleasant, like a bee sting, right? You don't intentionally go up and stick your hand in a beehive. Don't do that with jellyfish either. If you do get stung by a jellyfish, do not pee on it. The best thing to do is just wash it with seawater if you can. And if you have vinegar, vinegar also can help, but you don't want to create more of those stingers to be triggered, which is a common thing that people do by mistake.
All right. Well, thank you so much. That was a really fun one.
You're welcome.
And finally, Gabriella. I think that this one, for me anyhow, was the hardest to understand. So I want to congratulate you on kind of breaking it down for me. So let's see. First question, does a protein shape tell you everything about its function? So let me give you there's a couple of these. So does a protein shape tell you everything about its function? Or are there things about the protein that tell other things about the protein that tell you about its function. Also, you were talking about understanding how the protein moves, but aren't the proteins frozen when they're in the electron beams that they're going through?
Yeah, so I'll answer the first two first. So shape tells us most of what we need to know, actually. It's just that we really need to get into sometimes the like itty bitty details of that shape, like how that shape might interact with other things in the environment. But the shape is really the big determiner, yeah. For the frozen, that's a great question. So what's great about freezing our proteins is that it locks them in one position. But remember, we have millions and millions of copies of our protein in that single drop, and we freeze it in like flash freeze it. It happens super, super quick. And so the protein could have been doing whatever it wanted when it was in the droplet before we froze it. So that means it could have been waving around in all different ways, and we can capture it in different in all those different snapshots, kind of like we did for the horse or the Pegasus.
So it seems like proteins are kind of always moving, that they're just like kind of nonstop. What are they interacting with to do their jobs?
It's a great question. So they can interact with other proteins, both in proteins that are in our body, but also proteins of invaders. So like viruses and bacteria also have proteins, and a lot of the time what our body recognizes about them and how we figure out that they don't belong in our body is because they have proteins on them. And those proteins don't belong in our body, notices that, and then triggers our immune system. They can also interact with smaller chemicals. So even though proteins are really small, they're not the smallest thing in our body. So proteins interact a lot with like individual water molecules or smaller chemicals like drugs. A lot of drugs actually interact with proteins.
That was that image of the cargo, kind of. So I was wondering, like, what's the cargo?
Yeah.
That was a good image. So last question. You talked about how like the technology we need to see proteins is so advanced. Does that mean that we kind of only very recently discovered proteins? Because I feel like it's something that I have, grown up hearing about and like kind of learning generally in science about. So took it for granted that maybe we had known about them for a long time.
So unfortunately, I can't tell you when we got the structure of the first protein off the top of my head. And you're right that the electron microscope is actually a more recent invention, and it's something that honestly, only recently it got the Nobel Prize in 2017, and that was really when it started to like rocket off and be used for proteins. Before that, we'd used actually a different technique that involves X-rays. And that technique is also really, really good, but it has its own drawbacks. And so that's why there's kind of been this shift to electron microscopy, because it can identify and like fill some of the gaps that the X-ray technique can't use.
All right, well, I want to thank you all so much for presenting tonight. It was all really excellent and informative. And I want to thank the audience as well. Thank you for your questions. Thanks for joining. Our next UW Engage Science series event is going to be next week Monday, so tune in then. And I want to wish you all, the best of luck in your future studies and wherever you're at in them. Thanks so much for all the work that you do and for breaking it down for us tonight.